Skin functions as an exterior surface between the human body and the environment, acting as a physical barrier to prevent the invasion of foreign microbes while providing a home for the commensal microbiota. The harsh physical landscape of skin, particularly the dry, nutrient-poor, acidic environment, also contributes to the adversity that pathogens face when colonizing human skin. Despite this, the skin is colonized by a diverse microbiota. Our skin is completely coated in microbes. There are approximately 1 million bacteria composed of hundreds of species on every square centimetre of our body. Our skin is home to millions of bacteria, fungi, and viruses that compose the skin microbiota. As the largest organ in the human body, the skin is colonized by beneficial microorganisms, but when the barrier is broken or the balance between commensals and pathogens is disturbed, skin disease or even systemic disease can result. Studying the composition of the microbiota at different sites may be valuable for finding the cause of most skin disorders, such as acne, as well as eczema, dermatitis, or psoriasis. Structurally, the skin is composed of two distinct layers, the epidermis and the dermis. The outermost, the epidermis, is comprised of layers of differentiated keratinocytes. The top layer of the epidermis, or the stratum corneum, is composed of terminally differentiated enucleated keratinocytes, also known as squames, that are chemically cross-linked to fortify the barrier of the skin. Now, a keratinocyte is a cell of the epidermis that produces keratin. Sweat glands are important for the maintenance of body temperature through the evaporation of water, which also acidifies the skin, making conditions unfavorable for the growth and colonization of certain microorganisms. Sweat contains antimicrobial molecules such as free fatty acids and antimicrobial peptides that inhibit microbial colonization. Connected to the hair follicle and denser in oily sites, the sebaceous glands secrete lipid-rich sebum, a hydrophobic coating that lubricates and provides an antibacterial shield for hair and skin. The type of microbial communities on the skin is dependent on the physiology of the skin site, with changes in the relative abundance of the types of bacteria associated with moist, dry, and sebaceous microenvironments. Highly sebaceous sites, such as the face, the chest, and the back, are dominated by lipophilic Propionibacterium species. So lipophilic means having an affinity for lipids, such as fats. Bacteria that thrive in humid environments, such as the Staphylococcus and Corynebacterium species, were most abundant in moist areas, including the bends of the elbows and the feet. Fungal community composition is similar across core body sites, regardless of its physiology. So if you have ever wondered, as I did, why you only tend to get acne on the face, chest, and back, and not on the arms, legs, or lower torso, it's likely because the face, chest, and back are sebaceous sites dominated by the acne-related Propionibacterium species of bacteria, and it's the Cutibacterium acnes that's a member of the Propionibacterium species. Now, fungi of the genus Malassezia predominate on the core and arms, whereas foot sites are colonized by a more diverse combination of Malassezia, Aspergillus, Cryptococcus, Rhodotorula, Epicoccum, and other species. Compared to the intestines, the skin lacks many nutrients beyond basic proteins and lipids. To survive in such a cool, acidic, and dry environment, our skin's resident microbiota has adapted to utilize resources present in sweat, sebum, and the stratum corneum. C. acnes, for example, can thrive in the anoxic, which means greatly deficient in oxygen sebaceous gland, using proteases to liberate the amino acid arginine from the skin proteins and lipases to degrade the triglyceride lipids in sebum. This releases free fatty acids, which promote bacterial adherence. Sebum levels on the cheek are abundant with Propionibacterium species. The Malassezia species of fungi is the most abundant fungal organism on the skin. It coexists with C. acnes and other bacteria. Malassezia is the fungus thought to cause fungal acne. One study showed acne lesions were significantly reduced after administration of antifungal drugs. The authors suggested that Malassezia and not C. acnes was potentially the cause of acne not responding to treatment. The findings from several other studies support this hypothesis. One study reported that Malassezia restricta and Malassezia globosa can be isolated from young acne patients. Another study showed the lipase activity of Malassezia is around 100 times higher than that of P. acnes. Malassezia can increase fatty acid production, increase skin cell development in the hair follicles, potentially leading to a blockage and increase inflammation via activation of an immune response. It's the immune response that causes the acne lesion's redness. How much Malassezia levels are affected on the skin via the gut-skin axis is unclear, but it's not beyond the scope of possibility that a Malassezia overgrowth in the gut may be capable of relocating from the gut to the skin via the gut-skin axis. It's important, therefore, to optimize your gut microbiome, which is covered in chapter 6 of my book, whether you have bacterial or fungal acne. Looking at the gut-skin axis, chronic skin conditions such as acne are complex and affected by many factors. Current evidence offers insight into how the skin and gut microbiome are connected. There is evidence the gut microbiome influences other organ systems, and it has a particularly complex connection to the skin. Commensal or friendly bacteria in the gut may help modulate systemic immunity and reduce inflammation, which has an impact on skin health. For example, short-chain fatty acids are produced by commensal gut microbes when they ferment dietary fiber. Short-chain fatty acids regulate the life cycle of immune cells and have a protective role against the development of inflammatory disorders. This is relevant as acne has an inflammatory component. Another short-chain fatty acid, butyrate, can positively modulate the activity of inflammatory cells such as cytokines and suppress inflammatory immune responses and therefore acne. Under certain conditions, commensal bacteria can undergrow and pathogenic bacteria and other microbes can thrive. This creates a dysbiosis, which is an imbalance in the amounts and types of bacteria in the gut, which can have a negative impact on your health. Chemicals produced by these gut microbes may enter the bloodstream, build up in the skin, and impair skin health. This may occur due to the increased gut permeability, otherwise known as a leaky gut syndrome. If you have a healthy, diverse gut microbiome, it greatly increases, if not ensures, your health. Research has suggested a dysbiotic gut microbiome leads to disease and may be associated with skin disorders. Rather than addressing the skin alone, it's crucial to consider the role of the gut microbiome in skin conditions, including acne.